So I'd like to talk to everyone today about understanding cytokine networks, and in particular, the case of interleukin 2 or IL-2. This has been a topic that we've been working on since we started up the lab 12 years ago now, and we're continuing to work on it since we moved over to the Babraham 2 years ago. So interleukin 2 is a small secreted growth factor, first identified as a T cell growth factor. In vitro, you can add it onto T cells, the T cells proliferate and grow. It was cloned in 1982 and approved for clinical use in 1990. So this is a very old interleukin. For me, if it was cloned while I was in primary school, I'll consider it to be ancient history. Yet we're still learning a lot about IL-2, even after it's been in clinical use for 30 years. There's a lot of potential in cancer because of its pro-inflammatory effect. There's a lot of potential in autoimmunity because of its anti-inflammatory effects. Literally thousands of clinical trials using interleukin 2. But the actual clinical use of IL-2 is still quite niche because of a narrow therapeutic window. And this narrow therapeutic window is because of some unexpected side effects, the most common of which is eosinophilia. Now, just to get a grip on the complexity of IL-2, is you can see straight away that I've noticed this has got pro-inflammatory effects and anti-inflammatory effects, and you want to use it in diseases as diverse as cancer and autoimmunity. And of course, these can't both be true at once. The reason this works is because interleukin 2 has got multiple different receptors. The high affinity receptor is a trimer between IL-2 receptor alpha, beta, and gamma. The IL-2 receptor alpha I'll often call CD25 because that's its official name. It's a very high affinity receptor. If you don't have CD25 or the alpha chain, you have an intermediate affinity receptor. And if you just have CD25 or the alpha chain, you've got a low affinity receptor. Now, the reason why we're getting different effects for pro-inflammatory and anti-inflammatory is that if you're using a low dose of interleukin 2, you can feed the high affinity receptor, which is mostly expressed on regulatory T cells, which are anti-inflammatory. Whereas if you are using a high dose of interleukin 2, you can also feed the intermediate affinity receptor, which is expressed on inflammatory cells, such as CD8 T cells and NK T cells, and NK cells. So you are getting different types of response based on the dose, because the dose dictates which receptors can be activated. Now, I want to start by talking about a story that we started up, James and I started up 12 years ago when we first set up the lab with one of our first PhD students, Wim Pearson. And this is to work out how interleukin 2 is feeding regulatory T cells. Because it's a very important negative feedback circuit. So even though this story goes back a few years now, I wanted to set the scene for some of the more recent work that we've been doing since moving to the Babraham. Now, we're able to look at regulatory T cells using a FOXP3 reporter. FOXP3 is a transcription factor that's expressed in all regulatory T cells. And so by using a reporter, we can follow or manipulate regulatory T cells. And we had at our disposal two different reporters. One of them was Thy1.1, where we're expressing this marker under the control of the FOXP3 promoter. And a second is diphtheria toxin receptor, where we are expressing the receptor for diphtheria toxin under the control of the FOXP3 promoter. Now, fortunately for us, FOXP3 is on the X chromosome. And because of X chromosome inactivation, if you're a female mouse and you have both of these reporters, you actually have two Treg populations, one expressing one reporter, one expressing the other reporter. So in a heterozygous female mouse, we have 50% of the Tregs will be diphtheria toxin receptor sensitive because they're expressing FOXP3 DTR. And if we inject diphtheria toxin, we can wipe out this half of the population. And the other 50% of Tregs will be Thy1.1 positive, but completely resistant to diphtheria toxin. So this lets us wipe out half the Tregs at once and then look how the other half respond to that change in population size. So this is really the key figure from Wim's PhD. And what we're looking at here is the proportion of Tregs that are present in the spleen. This is as a proportion of CD4 T cells. And normally here we have about 5–6%. If we just look at the black line, this is the total number of Tregs. We can then add diphtheria toxin and the number halves very quickly. And then over the next few days, we start to see this rebound. And very quickly, you have a rebound far higher than the original population size. And then it takes quite a long time to dwindle back down to its normal size. Now, when we look at the DTR positive Tregs, these Tregs go down to almost zero after being injected with diphtheria toxin, and they take a very long time to recover. So the recovery is not because of a new wave of Treg formation, which would be split 50-50, but rather the surviving Tregs, they are expanding up in number. And I'll get to this later, but they're increasing their proliferation, they're decreasing their apoptosis. So you have a very rapid expansion, you have an overexpansion, and then you have a very slow contraction. If we look at what is happening during the expansion time frame, we see that there is a sharp increase in the amount of IL-2 produced by non-regulatory T cells. This is a reporter now for IL-2 production, and you see just a few days after diphtheria toxin receptor injection, we're starting to get an escalation in the amount of IL-2 being produced. So as soon as you don't have enough Tregs in the system, the non-Tregs, they start to produce extra IL-2. They now have permission to produce extra IL-2 because that suppression is reduced. We're now looking here at the response of the regulatory T cells to losing half of the population. So what we've got in each case here is the white bars are the Thi-1.1 positive Tregs. So these are the resistant Tregs. The black bars are the DTR positive Tregs. So these are the Tregs that we can delete. They're normally 50-50. If we deplete with diphtheria toxin, you see a collapse in the number of the DTR positive Tregs and a large increase in the number of Thi-1.1 positive Tregs. However, if we also add anti-IL-2 to block the extra IL-2 that's being produced, we see that there is very little expansion in the remaining Tregs. So that extra IL-2 is driving the increase in Treg numbers. We can see on the middle panel that this is caused by extra proliferation because the proliferation of Tregs, which is normally already quite high, jumps up to about 60% of Tregs if you have a niche filling situation. And we can impede this by adding anti-IL-2. And on the third panel, we're looking at the apoptosis rate, which is already quite high for Tregs. This drops down if you delete half the Tregs, because the Tregs at the left, they are filling up and they're not dying as fast. And this drop is negated by adding anti-IL-2. So I'm going to skip over a lot of molecular biology and a lot of mouse strain crossing to just give you the take-home message on what's happening at a molecular level. And basically, Tregs are primed to proliferate and die very quickly. They have this pro-apoptotic protein called BIM, which impedes MCL1, which is anti-apoptotic. If we have extra IL-2 in the system, IL-2 increases the expression of MCL1, and that blocks the apoptosis pathway. So that is the mechanism by which the IL-2 is allowing the Tregs to expand. This gives us a very nice negative feedback circuit. The Tregs, they need to have IL-2 to survive. However, IL-2 is limiting, and IL-2 is only coming from conventional T cells because the paradigm is that Tregs can't make IL-2, although we'll challenge this in a little bit later. They can't make IL-2, so they're dependent on IL-2 from T conventional cells or conventional CD4 T cells. In return, the Tregs are suppressing the production of IL-2 by the conventional T cells. So this sets the whole system in balance, where there's a very fine set point of the number of regulatory T cells and the number of IL-2 that can be produced in the system. Now, while we do have a balance here, this balance can be temporarily broken. If you've got an infection and you have an activation of conventional T cells, these conventional T cells are expanding up so that they can fight the infection. However, during expansion, they are producing extra IL-2. The extra IL-2 feeds into the Tregs. Those Tregs now expand and they start suppressing the expanded conventional cells. This causes a IL-2 contraction and shutdown. IL-2 becomes limiting again, and Treg numbers shut down, and you get back into homeostasis. So this is a system that has a negative feedback cycle, giving you a nice balance between regulatory T cells and inflammatory T cells. It does allow you to have an inflammatory response, but that inflammatory response is self-limiting. It shuts itself down, and that's a very important aspect of immune responses, which is all programmed into the immune response by this dependence of Tregs on IL-2. Now, one of the things I'd like to note here is that this system basically depends on the preferential feeding of IL-2 into regulatory T cells as opposed to inflammatory T cells. And this happens because the regulatory T cells are expressing the high affinity trimer, because unlike inflammatory T cells, they express CD25. Now, the second part of the story I want to get onto is from a really talented postdoc we had in Leuven, Stephanie Humblot-Barron, who is now an independent professor in Leuven. And together we worked on this rare disease, familial hemophagocytic lymphohistiocytosis, or FHL. FHL is a very severe disease, onset about 1 year of age, extremely severe inflammation and anemia. When onset occurs, there's normally a matter of hours or days before the patient will die if they're not treated very fast with very heavy immunosuppression. Now, 30% of these cases are due to mutations in a gene called perforin, which is a gene that inflammatory CD8s and NK cells use to kill off infected cells. So these are patients that have got poor viral clearance, and for some reason, this drives them into a severe inflammatory state with anemia. Now, fortunately, there's a very good mouse model of FHL, and this is a knockout of the perforin gene in mice. These mice are completely healthy until you challenge them with an infection with LCMV, which is just a quite an inflammatory virus. Now, if you have a wild type mouse and you give them LCMV, you get transient inflammation and you get viral clearance. However, in a perforin knockout mouse, you challenge them with LCMV, they get inflammation, but they also have impaired viral clearance because they can't get rid of all of that virus, and they end up having lethal anemia and inflammation. So they have the same genetic defect. They also need a viral trigger, which seems to be true in the patients, and they have very similar symptoms to the patients, the combination of anemia and lethal inflammation. The other thing I would note is that this is a pathology caused by the immune system and not caused by the virus. Because if you take a rag knockout mouse that has got no T cells and no B cells, you have a massive impairment in viral clearance. But the mice are surviving the infection of LCMV. They just have high virus titers, but they're not dying of the viral infection, which means that in the perforin knockout mice, it is the immune response against the virus that is killing the mice, rather than the virus itself. What is happening in these mice? Well, here we can take the wild type mice and the perforin knockout mice, we can give them LCMV, and we can look at the nature of the immune response that is generated. And the most obvious thing you see is an enormous response of interferon gamma. If we're looking over at the flow plots on the top left, you see in the wild type mouse, a very strong interferon gamma response. This is actually a very strong response because LCMV is a very inflammatory virus. By contrast, the perforin knockout is an order of magnitude stronger. It is just off the charts, the amount of interferon gamma that can be produced by these mice in response to an LCMV infection. And if we look at CD8 subsets at day 0, everything's completely normal. But by day 10, almost every CD8 cell is an effector memory cell in the perforin knockout mouse. This is incredible levels of activation of CD8s, and in particular, production of interferon gamma. So the model that was present before we started working on this was that LCMV in the perforin knockout mice triggers excessive activation of CD8s, and this interferon gamma causes the anemia and the lethal inflammation. However, when we started looking at these FHL mice, we saw that interferon gamma was not the only phenotype that was going very badly wrong. What we also saw is a collapse in the Treg numbers. So now we can look at the number of regulatory T cells present either in the wild type mouse or a perforin knockout mice. Without an infection, it's completely normal. Day 5, everything's normal. Day 10 after the infection, we have lost almost every regulatory T cell in this mouse, complete collapse of the population. And part of the reason why is the IL-2 consumption. Now, this is a wild type mouse if we're looking at the flow plots. And here we're looking at the expression of CD25 on the y-axis. And you can see in regulatory T cells, you have got a very high level of CD25. And on CD8 cells, most of these cells are negative. You've got a scattering of cells that are positive. And this is actually a very strong response because these CD8s are highly activated and are turning on CD25 at low levels. But this is about as good as CD8s normally get. If we look at a perforin knockout mouse, though, the story is completely changed. Regulatory T cell numbers, as I mentioned before, are very low. There's a slight dip in the CD25 expression on these regulatory T cells. But really, the thing that's off the charts is the expression of CD25 by the CD8 T cells. And here, almost every CD8 T cell is expressing just astronomical levels of CD25 to the point where the CD25 expression, the high affinity IL-2 receptor expression, is higher on CD8s than it is on Tregs. And we can show through downstream signaling experiments that this really inverts the consumption hierarchy so that IL-2 feeds the inflammatory CD8s instead of feeding the regulatory T cells. We can then formally test the hypothesis that the Treg collapse is important for disease. And what we did is a series of experiments in mice and humans. I'll start with the interferon gamma test. What we did is we took the perforin knockout mice, which can get FHL. We also crossed in interferon gamma knockout. And now when we gave them LCMV, we completely cured the anemia. So in fact, the anemia is due to the extra interferon gamma. But we did not improve the weight loss, we did not improve the inflammation, and these mice were still dying from a lethal inflammatory response. So in fact, that lethal inflammation cannot be due to interferon gamma in the mice. We were also able to identify a patient with FHL or HLH, which is a very similar condition with slightly different molecular basis, in a patient that had an interferon gamma deficiency. So this is a patient that has a complete loss of function of interferon gamma receptor 1, and they developed FHL after an infection with Mycobacterium. They had the hyperinflammation that we characterize with FHL, but they did have a reduced and atypical anemia. So we can say that the anemia does appear to be due to interferon gamma, but the lethal inflammation is not. To test the other side of the hypothesis, we were able to cross in a mouse that we generated where you can't turn on CD25 in CD8 T cells. So what we've got is CD25 flox mice. We crossed, which we generated, we crossed these to CD8 Cree mice so that now CD8 T cells can't turn on CD25. And then we crossed that to the perforin knockout. Now we inject these mice with LCMV, and we saw that we restored the Treg numbers, we reduced the weight loss, we reduced the inflammation, but we weren't able to have any impact on anemia. So this does demonstrate that that fatal inflammation is due to the IL-2 theft from CD8s, but the anemia is not. The anemia is due to interferon gamma. And if we look at a cohort of patients that were coming through our hospital in Leuven with FHL or HLH, we were able to characterize the number of Tregs that were present in these patients. There's quite a small number, and we're only able to get samples sometimes only an hour before treatment, sometimes a few hours after treatment, because as I said, this disease moves extremely quickly once it's triggered. Of the 3 patients that we had samples from before treatment, they all had Treg numbers that were well below the normal human range. And of the 4 patients that we were able to get hold of after treatment, 3 of those patients had restored themselves to the normal range for Treg numbers. And 1 patient was still under the normal range, and that patient died the next day. So this does to us demonstrate that Treg collapse is a part of the pathogenesis of FHL or HLH and is caused by the theft of IL-2 from Tregs being diverted into CD8 T cells. So when we talk about the pathogenesis of the disease, we do indeed have a defective viral clearance. We have an excessive activation of CD8s, and we do have an excessive production of interferon gamma, which leads to the anemia. But the lethal inflammation is actually a separate phenotype caused by the increase of the high affinity receptor for IL-2 and the theft of IL-2 causing a Treg death, and that then causes the lethal inflammation. So if we get back to this model of a negative feedback circuit, this is how everything should work, and it works most of the time. However, this negative feedback circuit can be rerouted into a positive feedback inflammatory disease situation. So here, the system that is nicely in balance, these CD4 T cells, if they get activated, they'll start to produce IL-2, they'll start to produce an inflammatory reaction. However, if the CD8 cells are excessively activated, and if they're able to express levels of CD25 higher than Tregs, the IL-2 that should go from the CD4 cells back into the regulatory T cells will be diverted. And so the regulatory T cells start dying from IL-2 starvation, while the CD8 T cells, they start getting boosted. And this provides an auto-inflammatory loop that goes on and basically causes a toxic inflammatory shock. So for that, I'd like to move on to the third and final part of the story about the IL-2 cytokine network. And this is a project that was started up with me and James a few years ago and really pushed forward by Carly in those last couple of years. And essentially, we wanted to look beyond this central circuit for IL-2 and start to understand what are the other things that IL-2 can do. What is it doing in homeostasis? How is the circuit running? Because if we want to start interfering in this circuit in a therapeutic sense, we do need to understand every part of the circuit and not just the central bits that have been studied more often. So if we start where we were at a few years ago in terms of the cytokine network, this was, I think, the basic understanding of how IL-2 is working. The main production of IL-2 is from CD4 T cells. CD8 T cells can also produce a little bit. This IL-2 is then feeding the regulatory T cells and the CD8s, but with preferential feeding to the regulatory T cells. They then block the production of IL-2, so you do get this negative feedback circuit. Some work by Carly, though, and this is, we just put this on bioRxiv a few weeks ago, so if someone wants to read it, they can search for it online. So that there is more diversity of IL-2 producers than we had previously, we were previously aware of. So if we look in the spleen and the lymph nodes, certainly CD4 cells are the main producers. This is the percentage of total IL-2 producers that are of different lineages, CD4 T cells, CD8 T cells, the vast majority. However, if we do look at tissues such as the lung, we start seeing other lineages come forward. And if we look under inflammatory conditions, we can see that minor populations that don't normally express much IL-2 start to become a more important source. So there is more of a diversity of producers than we expected. We also have some work by Kalish in the lab, who's looking at the diversity of IL-2 responders. And here what we've got is a stacked chart looking at the high affinity receptor expressors, which is in blue, the intermediate affinity expressors, which are in red, and the low affinity expressors, which are in white. And if we look in, say, the spleen, we can see that the CD4 Tregs, most of these cells express the high affinity receptor, while the CD8 conventionals next to them, they are almost all expressing the intermediate receptor, but there is a diversity of responders. And if we start looking again in other tissues, such as the lung, we see an increased diversity of responders. So the network has the potential to be more complex than previously assumed. So what me and James did is we started talking about how we could try to reroute the production of IL-2 to different parts of the network and see what this perturbation will do to the whole immune system. So what we came up with was a genetic switch that allows us to turn on IL-2 in different cell lineages. We have a ROSA26 promoter, which is a weak promoter, which is ubiquitously expressed. So if we turn it on, it'll be on everywhere. We then have a stopper, which is floxed, followed by an IL-2 cDNA cassette. So this means that we can now turn on a weak level of IL-2 expression in any cell type we want by using the appropriate CRE. Now, I really do want to emphasize that we are turning on a very low amount of IL-2 expression. We are not overexpressing IL-2 at all. If we look at the facts plot below, we see the amount of IL-2 that can be made by CD4 cells in the dashed line. And this dashed line shows quite a high production of IL-2 on stimulated CD4s. Compare this to the amount of IL-2 that we get from our transgenic expression. So now we have CD4 CRE ROSA IL-2, so that the CD4 cells are being licensed to produce IL-2 under the ROSA promoter rather than the IL-2 promoter. And you see a shift, which is about 100 times weaker than the expression that you normally get from a CD4 cell that has been stimulated. Here's on the right is now a graph looking at the natural expression. The expression on the first three columns are natural expression by CD4 conventional cells, Tregs and CD8s following stimulation. And then the expression driven by the transgene on the right. And you can see again that this expression is about 100 times lower. So we're not overexpressing, but what we are doing is we're turning it on in all of the cells at a low constitutive level. So it now becomes independent of stimulation. And this CRE switch allows us to target not just T cells, but any other cell type that we want to see what happens when they start producing IL-2. So this is the point where Carly came onto the project and really pushed this project forward. We started by looking at what happens if we get IL-2 produced by the normal sources, CD4 T cells, CD8 T cells. So we crossed the ROSA IL-2 to two different CREs to start with. There's the CD8 CRE, where IL-2 is produced by all CD8 T cells, and the CD4 CRE, where IL-2 is produced by all T cells. The difference between these is due to the point at which the CRE becomes active during T cell development. And essentially, what we see if we're turning on IL-2 in either CD8 T cells or all T cells is a very similar phenotype. And this is a massive expansion of CD8s. This first plot is looking at CD8 population from the normal to switch on with IL-2 in CD8s or in all T cells, huge expansion of CD8s. And in particular, these are the effector memory cells that are expanding up. The conventional CD4 T cells, they're actually collapsing. So for all that we're calling this a T cell growth factor, the CD4 T cells do not like the IL-2 at all. And the CD4 conventional cells are just collapsing in number, whether or not they are receiving IL-2 from CD8 cells or they're also producing their own IL-2. However, the regulatory cells, which are also CD4 positive, but they're not conventional, these cells are massively expanding as a population. And so we're getting to a point where almost every CD4 cell that's left is a conventional cell, is a regulatory cell rather than conventional cell. On top of this, these mice now have a fatal lymphoproliferation. Normally, lymphoproliferation is controlled by regulatory T cells. However, that control is partially because the regulatory T cells are able to shut down IL-2. In this case, they can't shut down IL-2 because that IL-2 is now made by a transgene expression, that is constitutively active. And so the lymphoproliferation can't be controlled and the mice start dying relatively early on in age. Now I want to spend a bit of time on this experiment because it's a really beautiful experiment that Carly set up. It's a little bit complicated though. So what we've got is a mixed bone marrow chimera, my favorite technique. And we're mixing the bone marrow from CD4 Cre Rosa IL-2 mice. These are mice where T cells can make IL-2 with wild type mice. And we have a congenic label, so we can spot the difference between the different bone marrows and also between the mouse that we injected the bone marrow into. And rather than doing a mixed bone marrow chimera at 50-50 ratio, we used lots of different ratios from 0-100 all the way through to 100-0. And what we've got on the plot to the left is a measure of the number of conventional T cells or number of regulatory T cells versus the ratio of the input bone marrow. And this input bone marrow goes from 0 being basically we're putting in wild type bone marrow to 100 being us putting in purely bone marrow where the T cells can make IL-2 and everything in between. So the way to read this plot is if we look at the light blue line, which is Treg numbers, we see the Treg numbers start out normal at 5%. Sorry, the Tregs are on the other y-axis, so they're at 20%. And as we increase the amount of IL-2 producing cells in the system, they are increasing up until they're reaching this plateau at about 70%. So the Tregs are increasing with the more IL-2 production that is present. The same is basically true for CD8s, while CD4s are decreasing with the more IL-2 production that is available. So this gives us a very similar result to what our previous analysis was. The CD8s and Tregs expand while the CD4 T conventionals collapse. The really interesting thing though, is if we're looking at the ratio of the different bone marrows compared to the ratio of input. So here what we've got is whether or not the expansion caused by IL-2 is happening in the cells that can make IL-2 or the cells that are not making IL-2. Here in black, we have B cells, which basically just shows that our input ratio is very similar to our output ratio. And then essentially, if the line is above the curve, this means that there is a preferential advantage for the cells that are making IL-2. So above the curve, the CD8 conventionals, that are making IL-2, those ones are preferentially expanding. And in fact, they're almost 100% of the cells that you recover are the CD8 cells that can make their own IL-2. So the CD8s love making IL-2 themselves. The CD4s and the Tregs, they do not like making IL-2. They are below the curve. So even though the Tregs increase with more IL-2 availability, the cells that can make their own IL-2, they're not the cells increasing. It's the cells that don't make the IL-2. They like seeing IL-2, they don't like making it. And same for the T conventionals. Not only do they not like seeing IL-2, they in particular do not like making IL-2. So we wanted to work out what's happening with this. Why don't CD4 T cells, in particular CD4 T conventional cells, why don't they like making IL-2? They should at least be neutral to it, even if they can just ignore it. But instead, it actually seems to have a toxic effect on production. So what we did here is we looked at a number of different cytokine pathways, and in particular, the IL-7 pathway, which is known to be very important for the survival of conventional CD4 T cells. They really do seem to be dependent on how much IL-7 is present in the system. The receptor for IL-7 is CD127. So we can look at the expression of CD127 as an ability of the T cells to see IL-7. And what we've got here on the first panel are wild type cells or cells that are making IL-2. And the conventional T cells, the CD4 conventional cells that are making IL-2, these cells actually have a lower level of IL-7 receptor, whereas CD8s, their IL-7 receptor is actually going up. We can also look downstream of the IL-7 receptor to phosphostat 5, and the activation of phospho-STAT5, so the activation of the IL-7 pathway. And in particular, if we look at the naive T conventional and the activated T conventional cells, the amount of phospho-STAT5, so the amount of activated IL-7 receptor pathway, is going down in both of these cell types. So this means that the CD4 T cells that are making IL-2 have actually reduced the signaling through the IL-7 receptor pathway. And finally, in the third panel, what we did is we removed the ability of CD4 T cells to make, or actually of all T cells, to make IL-2. So we have CD4 Cre, which is active in all T cells, and then a floxed allele of IL-2. So these cells can no longer make IL-2 at all, whether or not this transgenic form or the endogenous form. And when we look at these mice, we see that the T conventional cells, the CD4 cells that can't make IL-2, they now have a boost in CD127. So it does seem like the production of IL-2 is actually causing a toxicity cost by making the cells desensitive to IL-7. And that appears to be driving the phenomenon that we just described. So if we go back to this IL-2 network, we start off with a very simple network that was kind of the dogma in the field. We're now able to add on a few extra parts to this network. One thing I didn't really mention, but this is some very nice flow by Oliver Burton in the lab, showing that regulatory T cells actually can make IL-2. They're often described as having a silenced IL-2 allele. We've been able to show that this IL-2 silencing is only partial, and they've reduced the expression of IL-2. So they are dependent on IL-2 from other cell types, but they haven't completely lost the expression. It was just that the staining other people were doing was not as sensitive as our staining. So we have extra sources of IL-2, such as coming from Tregs. We also have this production associated fitness cost, which is actually making this producing CD4 T cell less fit and having a toxicity cost. And we're able to show that CD8 cells, they actually prefer their autocrine sourced IL-2. So they have this preferential expansion loop. Just to touch on a couple of very, very short stories, one panel each. We were then crossing the Rosa IL-2 Cre on a Rosa IL-2 mouse onto a NK Cre, NKp46 Cre, which is now activating IL-2 in NK cells. And when we get NK cells to make their IL-2, the only phenotype we see is that the NK cells are massively expanded. So this is very similar to the CD8 story, where the NK cells really like an autocrine circuit. Rather than feeding into the common pool and feeding Tregs, they feed themselves when they produce IL-2. Whereas if we make dendritic cells produce IL-2 with a CLEC9A Cre, the dendritic cells, the only phenotype that we really see there is a very large expansion in the number of Tregs. So the dendritic cells, they are directly feeding the Tregs with their IL-2. The most interesting phenotype that we saw, though, was when we had IL-2 being made by B cells. Now, B cells are not a major source of IL-2 in a normal situation. Unless you have a parasitic worm infection or the like, you don't get much IL-2 from B cells. They can make IL-2, though, under those circumstances of a helminthic infection. Now, what we've done here is we've compared CD4 Cre to CD19 Cre. So either IL-2 being produced by all T cells or IL-2 being produced by all B cells. Now, if we look at the left-hand panel, we can see that IL-2 production in terms of the total production of IL-2 is very similar, whether or not it's produced by all T cells or all B cells, because you've got very similar numbers of T cells and B cells in the system, and then Rosa promoter has an equal strength in either cell type. But even though you've got a similar amount of IL-2, you have very different phenotypes that emerge. So I've just described the phenotype of CD4 Cre produced IL-2. You have an increase in Tregs and increase in CD8s. Very different response when B cells make IL-2. You have an increase in eosinophils and an increase in CD8 Tregs. So I'll just start first with the eosinophil response. On the right-hand panel, we can see the normal number of eosinophils in the spleen, which is next to 0. A couple of cells you start to see in a mouse that has got a CD4 Cre, activation of IL-2 by T cells. If you look though at the CD19 Cre, where IL-2 is produced by B cells, you see that nearly half the spleen is eosinophils. This is just chock solid full of eosinophils. Very strange phenotype. And it turns out that this is not because the eosinophils are responding to IL-2. That would just make the story a little bit too simple. No, it turns out that the B cell derived IL-2 is expanding a very rare cell type called ILC2s, innate lymphoid cells 2. These cells are almost non-existent in a homeostatic mouse. However, if you have CD19 Cre and B cells are making IL-2, there's now a sizable fraction of ILC2s that are present in the bone marrow, the spleen, and the lymph nodes. Now, critically, ILC2s are an excellent source of another cytokine called IL-5. And if we look at now the right-hand panel, we have a normal number of eosinophils. In the third column, we have B cells producing IL-2. So we have a big increase in the number of eosinophils. And now if we neutralize the IL-5 with an anti-IL-5 antibody, this collapses down to nothing. So it does seem like the B cells, their IL-2 is expanding up the ILC2s, and their IL-5 is expanding up the eosinophils. So there's several steps in the pathway. Now, just note that we see the same phenotype of eosinophils if we use CD23 Cre, which is present in mature B cells, but not in OSX Cre, which is present in the bone marrow. So this is about the production of IL-2 in peripheral B cells rather than in bone marrow cells. I just want to briefly go back to the very first slide that I had for IL-2. And this is that when you actually deliver IL-2 to patients, the unexpected side effects you see are almost universally eosinophilia. And we've never really had a great explanation for this. There have been some papers trying to say that human eosinophils might express IL-2, but they don't particularly respond to it in vitro. And now I think we can say that it's not a direct response of IL-2 for the eosinophils. Instead, it is an expansion of ILC2s. They're producing IL-5. The IL-5s are driving the eosinophils, the eosinophilia. And so I think that's a very interesting mechanism that by injecting IL-2, we are basically recapitulating the phenotype that we see when B cells make IL-2. So this brings me on to the final aspect of this story, which is the CD8 regulatory T cells, the FOXP3 positive, CD8 positive cells. Now this population is very, very rare, 0.1 to 0.3% of CD8s express FOXP3. It's been noted a few times, but they're almost impossible to study because they're just so incredibly rare. However, when B cells make IL-2, we're now seeing 20 to 30% of all of the CD8s are now FOXP3 positive. Colossal increase. And this doesn't happen if T cells make IL-2. It really has to be B cells making the IL-2. Now, the nice thing here is that we've got now a sizable population of CD8 positive, FOXP3 positive cells. So we can actually purify these cells and start to do suppression assays. And so this is a classical in vitro suppression assay on the right, where you are starting out with a very low number of regulatory cells and you're increasing it to a point where you've got one-to-one regulatory to effector cells and you're measuring how much suppression. And in black, you have the results that you'll see from CD4 Tregs. The more CD4 Tregs you've got, the more suppression you've got. And in red, we've got CD8 Tregs that we purified from these mice. And the curve is almost identical to the CD4 Tregs. So at least in vitro, these CD8 Tregs do look to be as suppressive as the CD4 Tregs. So they are really bona fide regulatory cells and not just FOXP3 positive cells. But why are they increasing with B cell derived IL-2? Well, what we did here is to go back and look at the rare CD8 positive, FOXP3 positive T cells that we see in the wild type mice. So this scattering of 0.1 to 0.3%. And we phenotyped these for a number of different markers. And what we saw is that the FOXP3 positive cells essentially all express this chemokine receptor, CXCR5. So we can see there's a spread of cells on conventional CD8s, but the regulatory cells, they're almost all expressing CXCR5. And this is quite important because CXCR5 is a chemokine receptor that allows cells to get into the B cell zone. And now by immunofluorescence, we can look at a follicle and we can see the B cell zone. On what we have below is immunofluorescence. The B cell zone is in white with IgD, and then we've just removed the IgD on the right-hand panel so that you can see it a bit more clearly. And there are not many CD8s in here because, of course, most CD8s are in the T cell zone. But if we do home in on the CD8 cells that are there with the green outline, we can see these cells that have the green outline, so they're expressing CD8, and then inside they're expressing FOXP3. So these are the FOXP3 positive, CD8 positive cells, and they're located in the B cell zone. And in fact, if we look at the CD8 cells that are present in the B cell zone, about 60% of these are found in are regulatory FOXP3 positive CD8s rather than conventional CD8 cells. So even though they might only be 0.1% of the total CD8 population, they're 60% of the population in the B cell zone. So now it's not so difficult to see why they're responding to IL-2 derived from B cells and not from CD4 T cells. This is because where they're located in the B cell zone, they're completely surrounded by B cells, and the CD8s are quite far away. And IL-2 has a very short half-life and is continually consumed by its surrounding cells. So IL-2 is only able to migrate between 50 and 100 micrometers before it is completely gone. So these cells are probably completely isolated from T cell IL-2, but the second they start getting B cell IL-2, they're able to proliferate up and expand. So if we go back to this IL-2 network, we can now add a completely novel half of the network, and this is the B cell side. And when B cells make IL-2, they're excellent at feeding these CD8 Tregs because they're co-localizing in the B cell zone. The ILC2s also border the B cell zone, and they're responding to the B cell derived IL-2. And in turn, they can feed eosinophils via IL-5. So we now have a much more complex view of the IL-2 network and how it can respond to IL-2 from different cellular sources, different contexts. And I think this is quite important because if we understand the complexity of the network of producers and responders, we can see that we have both major and minor circuits. Now, the major cellular circuit of Tregs and CD8s is absolutely the pivotal circuit. And this is the circuit that we tend to focus on therapeutically. And it is a circuit that is amenable to engineering receptor affinity, because in most cases, the CD8s and the Tregs are using different receptors. So by modifying the cytokine receptor binding, we can modify whether or not Tregs get it or CD8s get it. However, there are also minor circuits that I've spoken about that are anatomy dependent. And here, I think the lesson is going to be quite different because we're not reliant on different receptors. We're actually relying on anatomical segregation. So if we want to avoid or amplify these minor circuits, we need to engineer the homing of the IL-2 rather than the receptor binding of the IL-2. For example, if we want to avoid eosinophilia, we can't do this by editing the binding to the IL-2 receptor because the ILC2s use the same receptor as Tregs. The only way to do this will be to segregate the IL-2 out of the B cell zone so we're not feeding that circuit. With that, I'd like to finish up. This has really been a labor of love for the lab over the last 12 years, really. I'd like to thank a number of people from Leuven, especially Stephanie and Wim, and a number of people in Babraham, especially Carly, James, Klish, and Oliver. But really the whole team has put in and worked on this project over many years. Thank you.
Thank you very much, Adrian. This was a tour de force of IL-2.